Good morning, everyone. Hear me well? Cool. Thank you, Dave, for the invitation to join you all. Everyone for inviting us, myself and many of the others that you will hear from this morning. You heard from Dave, I'm a soil biogeochemist, so I figured I'll start by telling you about what the heck a soil biogeochemist actually does, right? Um, So in our lab at UC Merced, the soil biogeochemistry lab, we study mechanisms that are responsible for the earth system, the soil part of the earth system in particular, to impose important controls on the earth's climate. And that is by controlling the cycling of carbon and release of greenhouse gases into the atmosphere. In particular, in our lab, we study the mechanisms that are responsible for actually keeping carbon in soil, as opposed to it quickly decomposing and getting back into the atmosphere as greenhouse gases. So a lot of that work revolves around carbon, but we also study biogeochemical cycles of other essential elements such as nitrogen and phosphorus. Um, and a big part of the mechanisms that are responsible for stabilization of soil organic matter has to do with abundance and chemical bonding of the organic compounds with surfaces of reactive minerals in soil, which are products of extensive weathering, chemical weathering and physical weathering in the environment, i.e., extensive transformation of rock in its way becoming soil. And so, hence why we call this field nanobiogeochemistry, to highlight the fact that so much of that stabilization is actually made possible by nano-sized natural minerals in the environment. Um, so a lot of this, as I mentioned, has broad implications to climate-related work, but it's also critical for soil health because healthy soils have a lot of carbon, and soils that have a lot of carbon can hold on to a lot of nutrients, water, and so there's a broader conversation around soil health, preventing or addressing degradation of soils due to a variety of human actions. And we're not talking about this today, but we have another broader mission around political ecology, i.e., application of political economic theory to issues of human environment relations to help understand how in processes that lead to degradation of soil and the land environment overall, and the implications for that for human and national security type of considerations. Okay, so I'll start by telling you a little bit about how a soil scientist like me is obsessed with the world of soil, thinks about organic matter and why organic matter becomes kind of really important piece, and then talk a bit about the application of NMR in soil science. I'll start with a little bit of history, pretty general, and then give you two examples of how in our work we've applied NMR and how helpful it's been. And then end with some conversation about what are the remaining challenges that are quite tricky for people like me. Okay, so I think everybody's probably on the same page here, but just as a way of level setting, let's start with from a fact that the climate's changing, right? Partly because, well, largely because we human actions keep releasing on the order of about 11 petagrams of carbon to the atmosphere through a combination of processes that have to do with burning fossil fuels and changing the way we use land. And this has caused the global concentration of atmospheric CO2 to increase by more than half since the Industrial Revolution. So that's pretty bad. But I think it's important to realize that all the things that we're facing right now as a result of climate change, though, all of those are the consequences of 50% of the amount of greenhouse gases we've released into the atmosphere. In some ways, you can think about we're only facing the consequence of half of our actions. And that's because so far, the natural environments that has to do with land and the oceans are taking up half of the excess CO2 we keep releasing to the atmosphere through a process we recognize as carbon sequestration. So slightly different mechanisms, but largely revolving, especially on land, around plants. They grow, they take out CO2 from the atmosphere during photosynthesis. They use it to build their biomass, and upon the death of those plants or animals that consume the plants, that carbon finds itself in soil, right? And obviously, though, what's really important to recognize is that carbon comes into soil through photosynthesis, but most of that actually decomposes through the activity of microbes and goes back up into the atmosphere as greenhouse gases through the process of decomposition. And important to recognize that from a perspective of our biogeochemists, the soil scientists, and ecologists, decomposition is an extremely essential process, right? Because it's the process by which all the nutrients that plants took up from the soil when they were growing, it's the process by which they're released, made available in soil for the next generation of plants and microbes to be able to grow, right? But in the of carbon coming in and carbon coming out, the balance becomes important. So how much of that carbon every year goes out relative to how much came in into the soil becomes an important consideration. And historically, soil system has been able to store a lot of carbon because small amounts of that carbon that came in through photosynthesis was able to be stashed away, if you will, in a soil carbon bank. I like the bank analogy because it allows people to think about things coming in and out and that the balance being what's really important, right? Most people can relate with the fact that at the beginning of the month, we're all super rich, right? The check hits our bank accounts and our balance looks really great. But the reality is that's not how rich we are, right? We still have to pay for the expenses, and that's decomposition. The nutrients have to be released. What really matters for our wealth is how much we are left with at the end after we paid all the bills and all our expenses. And that's how we think about carbon cycling in soil and carbon storage in soil as being dictated by that balance at every given time point of how much coming in and how much is leaving the soil system. But over long periods of time, even a small amount of that carbon that's stashed in soil contributes to a large amount of carbon buildup relative to any other reservoir for carbon in the Earth system. And I'm going to take a little bit of detour now, tell you about soil organic matter in particular. So from a chemistry or material science perspective, what I recognize is that it's breaking up, but don't think it's me. What most of us recognize is the fact that soil happens to be the most complex biomaterial that we in the Earth system. And complexity is attributed to the structure and dynamics of soil organic matter. And soil organic matter being this heterogeneous mixture of formerly residues formerly living plants and animals in soil that exist at different stages of decomposition. So soil organic matter being, again, this heterogeneous mixture of residues of formerly living plants and animals that exist in soil at different stages of decomposition, plus living microbial biomass and products of microbial synthesis, in particular, the remains of microbes, and that we refer to as microbial necromass. Another really important fact that I want to make sure you appreciate is the fact that under most conditions around the Earth, carbon content in soil, carbon concentration in soil is less than 5% biomass. Most of the mass of soil is actually minerals. It's the broken down pieces of rocks that are the inorganic constituents that make up the sand, silt, and clay, right? But that 5% is largely what controls so many of the critical essential life processes that soil provides, including production of food, including the soil system serving as a food and energy source for biota, including plants and microbes, really controlling the ability of life to thrive in soil and buffer against rapid changes in pH, control availability of nutrients through a process we recognize as cation exchange capacity, because most of the plant essential nutrients are cations that have to be exchanged with the surfaces of minerals in soil, between surface minerals and soil solution. So all of that is really controlled by that less than 5% of carbon biomass. There's a few soils, they're largely the exception, that exist either in wetlands, mangroves, or in high latitude environments in the Arctic that actually do have a lot more than 5%. We talk about those as organic soils. By definition, if it has greater than 20% biomass organic matter, about 12% by biomass carbon, then we refer to those soils as organic soils, and they're actually in a whole different category of soils the way we study them. But most of the world has mineral soils that have less than 5% carbon. Then again, even though it's a small proportion, it controls largely all the critical biological, chemical, and physical processes that allow soil to support life in all the ways that we know of. Okay, and then The other really critical thing to remember is that, remember I told you over time, small amount of carbon accumulates in soil and contributes to buildup of the soil carbon stock or bank. And what that has amounted through history then is that now there is twice more carbon in soil than there is in all of the world's vegetation combined, plus what's in the atmosphere that we're all worried about with climate change. So add them up, twice over, there's still more carbon in soil than in any other reservoir, with the exception of the ocean, which has a pretty large stock of carbon. And what that means then, especially when you think about the fact that carbon in soil is constantly exchanging with the atmosphere, so it's a continuing cycling between the atmosphere, plant, soil system, right? So a small change in the rate at which carbon is taken up from the soil, from the atmosphere to the soil, or a small change in the rate of decomposition that release carbon from soil to the atmosphere leads to significant consequences with respect to the atmospheric concentration of CO2. And a really important other piece is the fact that carbon in soil cycles relatively slowly compared to carbon in plants, for example. Because in plants, plants take it up through photosynthesis. They rather quickly die within a few years for the most part. Some of them live a few decades. Very few of them live centuries, right? But almost all living plants on the surface of the earth, all living things really, live about a take max, and then they die. So that carbon goes back up into the atmosphere. But if carbon makes it into soil, in particular in these associations that are represented by this cartoon here, that is showing if it finds itself inside aggregates of minerals, which makes it harder for microbes to get inside those aggregate spaces to decompose carbon, or for water and oxygen to be able to actually diffuse into those spaces to facilitate aerobic decomposition, then carbon persists. The other way is that carbon persists if it binds to the surfaces of reactive minerals that we know it can do through a whole host of chemical mechanisms, bonding mechanisms. So when it does that, then it persists in soil for a long period of time. And if you want to think about it from the perspective of climate, for example, what really should matter, right, is not how much carbon we can put into soil through photosynthesis or whatever else, right? Really most is how much will stick around in soil, stay there so that it doesn't have to go back so fast to the atmosphere. For that, it's these mineral associations that are key. But there's so many still remaining fundamental science questions that we are trying to clarify and to get better understanding on how different types of molecules are stabilized in soil, for example, in association with minerals. What are the degradation pathways for different carbon-based compounds? All of that remains an active area of research that many of us that you'll hear from the discussions today work on. Okay, so let's switch to NMR. So NMR is a really neat tool that a lot of soil scientists and biogeochemists like to use and apply. And all of this, I think it's fair to say, started getting popular in the 1960s when people were compounds, so that these things that we call humic and fulvic acids that represented organic compounds that cycle the two different rates with soil, using some pretty strong acids and alkali solutions. They would extract them and then to do liquid state 13C NMR and every once in a while liquid state proton NMR. It became, I think it took hold, and it was a start of in some ways, a revolution, if you will, but it had limited utility because what we do know is that most organic matter is bound to mineral surfaces and is actually not easily extracted even with those harsh chemicals. And whatever you extract, almost kind of certainly at this point, is really bears the signature as an experimental extraction artifact more than actually tells you about the chemical nature of the organic matter in soil as it is. So we started recognizing, as soon as we were using these tools, there's a lot of folks started recognizing that these extraction procedures are really not helpful. If anything, they're creating misleading results. So there was a bit of a lull, if you will, on NMR on extracts. But in the 1980s, a whole kind of different group of people started picking up solid state NMR work with 13C in particular on whole soils that are not altered in any way without any extractions, which is exactly what we need, right? We were hoping to eliminate the need for those chemical extractions that altered the nature of the sample and the organic matter. So that really made it possible for people to think about, okay, we can start using NMR, at least solid state, direct polarization, but also cross polarization, and magic angle spinning to actually be able to get a sense of what is soil organic matter made out of, and how do those compositions change as a function of all sorts of processes in nature. Since the 1990s, I think there's been all sorts of explorations to, broadly adopt solid state 13C NMR, but even more kind of explorations into the world of 15N NMR, and further work on liquid state proton and P31 NMR, and a variety others that people use that I don't know. They're not necessarily more popular than these, but people use a lot now. And then there's even a lot of efforts to think about more advanced techniques with 2D high resolution NMR techniques. And there's a lot of excitement, I think it's fair to say, for people to be able to get a bit more granular view of the molecular components of soil organic matter without doing these destructive extractions that we know were not really helpful. Okay. So really the important focus for a lot of people have been then, how do we get this chemical information of soil organic matter without having to destroy the soil, without destroying the spatial and contextual structure. Oops, I just noticed a typo as I'm looking at this. Structure of the organic matter, and be able to actually describe it. Because soil is complex, as I mentioned, right? It's complex. It's made up of all sorts of things. And the minute you start destroying this, you've lost a lot of meaning, right? Even from the physical stabilization perspective, so much of the organic matter that's made that remains stable or persists in soil for a long period of time because of the structure, as soon as we destroy it, that some of that information is lost, right? Even as soon as we dry it and as soon as we grind it, let alone if we actually added harsh chemicals that don't really exist in nature and try to extract and interpret what we got as a result of that. So anyway, 13C NMR really took off because of its potential to provide good information of chemical signatures of organic matter without destroying the contextual information, at least to the degree that liquid state methods did. But I think a long list of concerns remain with how we use NMR. It's largely because of how heterogeneous the media we're working in is, right? And also a range of issues that are associated every time you think about doing NMR with soils. To start with, I mentioned most soils have carbon less than 5%, but the reality is a lot of soils in the world actually have less than 1% carbon. And so there's a very weak signal that you're trying to detect in a heterogeneous media to start with. And it's not just heterogeneous because there's all sorts of other things. There are things that actually make it exactly precisely harder for NMR. Things like iron, iron oxides, and a bunch of other paramagnetic minerals that cause complications with how we run and interpret NMR, causing line broadening, reducing relaxation times, spreading signal and signal and reducing resolution. And then, of course, I showed you earlier, I started out by saying organic matter is super heterogeneous. And in the context of NMR, then we're looking at a material that spans multiple different types of compounds, that some are incredibly rigid aromatic type of compounds, including char and pyrogenic carbon that I'm going to talk about later. Some are semi-mobile, the polysaccharide type of things, and then fairly mobile ones like lipids, and basically meaning that we now have to think about a media that analyze where no single pulse sequence can actually optimally detect all the different components simultaneously. So trade-offs basically that we have to work with. And then as you will see the spectra that I show you, they're never as clean as you all do in your world of chemistry, right? There's this overlapping spectral regions that make tricky, the really broad peaks that we have to work with, and we have to make peace with that. And we also have to make peace with the is incredibly variable. So the sample to sample variability is incredibly high, making it really hard to actually standardize different approaches. The one workaround, even for solid state, that a lot of people do, even we do sometimes, is HF demineralization to be able to remove some of the minerals, especially the paramagnetics, and then concentrate the carbon and make it easier for cross CP NMR at 13C NMR. But the problem here is, yes, it does that. But as I mentioned earlier, that most carbon is bound to minerals. So if you're dissolving the minerals, you're making the carbon that's bound to the minerals now be released into the solution phase. And while I'm rinsing out HF, I also lose all of that carbon, right? And so the recovery of some of the samples from some early work that we've did, actually when I was at Berkeley, can be something like 30, 40% of the original carbon you started out with. Yes, you're doing solid state NMR, but at some point you're wondering, what is the point if I don't even know what I lost, right? And so it remains a pretty, I just want you all to have these points in mind when I show you the spectra from some of the work that we've done. Okay, so let's talk about erosion. Erosion is a topic that I've spent a lot of time thinking about because it's one thing to think about how we characterize and understand soil in one place here and another place there. But if we're trying to connect it at a larger spatial scale, then we have to recognize that the nature of the landscape, the geomorphology of the landscape, the topography dictates that soil, associated organic matter, everything is moving laterally all the time in the Earth system. Some of it is moving naturally by wind or water. Some of it, the lateral transport of topsoil and associated carbon, is being made faster by human actions like tillage, of water, cultivating and using land improperly in areas that are not suited for cultivation or physical mixing practices like tillage that make it very easy for topsoil and associated carbon to move around in the Earth system. Some transport is rather water-assisted transport is pretty short term. So what typically happens with water erosion is that what happens with water erosion is that what you notice in the bottom part of the slide here, where over time, topsoil carbon that has higher percent carbon typically than subsoil gets mobilized over time and gets deposited downhill, just downhill within the same catchment that it came out from. But over successive transport periods, then the topsoil carbon that had a lot of the topsoil, the soil from the top part of the soil profile that had a higher percent carbon gets buried. So in a depositional setting, you have a lot of carbon with a lot of soil with a lot of carbon that gets buried. So, we've studied for a long time then to try and understand how topography and erosion and associated lateral redistribution of topsoil controls how long carbon can actually persist in soil. And this included work where we were quantifying the effect of erosion on the rate of inputted carbon, the rate of loss of carbon, the change in soil physical properties, in particular lateral redistribution of reactive minerals that play disproportionately high role in carbon persistence. And all of this work was to help explain how much of terrestrial carbon sequestration is actually induced by erosion. And this is a tricky topic to talk about, even in the context of soil science, because most of us are, if we heard about erosion and the concern associated with erosion, we thought about how erosion is destructive to soil, topsoil is lost, carbon's lost, nutrients are lost. All of those remain true, right? I just told you it's lost from one place, accumulates in another. What most soil science until the late 1990s had done, though, was focused on the top part of the hill slope where it was losing carbon and didn't really account for where that carbon was actually going and didn't recognize that that carbon was actually not leaving the catchment that it came out from. It didn't get delivered to the ocean. It's just downhill, being buried in all sorts of depositional settings. And also since the Industrial Revolution, human beings have built tons of depositional settings for eroded carbon in the form of reservoirs, artificial lakes, floodplains that we expanded, and all sorts of things that you can imagine. So on the order of about 4.5 gigatons of carbon every year was being moved and actually being buried downhill. And we were not capturing that in our global carbon and climate accounting. And once you capture for the potential replacement of some of the carbon that was lost by erosion on the hill slopes and its burial and stabilization, i.e. slower rate of decomposition in the depositional settings compared to the hill slope that it came from, then you actually realize that erosion, the process itself, actually causes more carbon to accumulate in the soil system than a world that operates without erosion. This was a really interesting thing that we've tried very carefully to explain, because no, we don't want anybody to come away from this talk thinking that erosion is a good thing. It is not a good thing in any way. But the way the world works, this mass transport, lateral distribution of mass actually causes for more carbon to persist on land longer than it would have done without that lateral redistribution. And so we can talk about that later. So this was one of the first projects where I was trying to use NMR to help understand the nature of the carbon in the hill slopes versus in different types of depositional settings. I had all sorts of quantifications about rates of input, rates of loss, change of physical properties, chemical reactivity. But we also wanted to see, is the chemical nature of the organic matter different in the depositional versus the depositional versus the eroding landform positions. So what you're seeing here is that this is within one small catchment, not far from here in Marin County, right close to the Golden Gate Bridge, a small zero-order catchment where we sampled different positions of it. These two are net eroding positions. This is at the very top, basically near the crest of that catchment. A summit, and then this is a backslope position, the fastest sloping part of that catchment. And these two are depositional settings. This is Marin County. Landslides are very common. So what this halo is a landslide scar from about 14,000 years. And soil has been accumulating for the last 14,000 years in this landslide scar that was scooped out, as I'm sure you've all seen the landslide scars, right? They look like you scoop them out with a spoon. And so over 14,000 years, sediment had been accumulating. And this is an alluvial colluvial plain, i.e. right at the bottom of the hill slope, where it captures all the material transported by gravity-driven transport, water, wind, everything else. And so two types of eroding positions, two types of depositional settings, and we went on to try and characterize what the nature of the organic matter looks like. This is all work done at the Pacific Northwest National Laboratories Environmental Molecular Science Lab in a pretty low field magnet. And we tried a whole host of DP techniques and other ways, because it was fairly early for them to use NMR on soils at that time, even though, of course, that facility, they do NMR on pretty much anything you can imagine, right? But we worked with folks there to adopt these techniques for soil. And we're able to get the spectra, which at the time was incredible, which was just not easy to get access to be able to do all of these and a lot more that I'm going to show you. And we get some really useful information, right? There's some really clear differences that show up. And in my world, we like to think about NMR results in particular in these two approaches, because what I'm trying to get is, first is the broader kind of fingerprint type of composition of organic matter. But then I'm specifically interested in, is this material transformed because of decomposition or not? So I compare the relative area under the curve for alkyl groups versus O-alkyls as a way of kind of detecting how much transformation that organic matter has seen. Presumably, if it's been decomposing a long time, the O-alkyls have been disappearing, relatively speaking, faster, i.e. the alkyls have been getting relatively enriched in the remaining organic matter. Same thing with aromatic groups. We expect that over time, as the material decomposes, more and more aromatics would be enriched in the remaining organic matter. So what becomes very clear is that, even if you just look nothing but just the overall change in alkyl to alkyl ratio and aromatics in the depositional settings, which you can easily identify here because those are the deeper ones, the soils on the eroding hill slopes are shallow. There's relatively small change in both that occurs in the depositional settings that compared to the eroding settings. And one of the messages that came out of here is that this is the pattern. If you follow the aromatic pattern here for the dark line, which is the top part of the eroding landform position, right? That's what I would have expected for most soils that are not experiencing kind of significant change because of lateral redistribution. Because I would assume that carbon in deep soil layers is more decomposed, so it has lost a lot more of the alkyl, O-alkyl groups, and it's enriched in more alkyls and aromatics. But if I look at what's happening with the depositional settings, though, relatively speaking, that yes, that happens. Of course, decomposition doesn't stop, but relatively speaking, it still retains quite a significant amount of all types of organic compounds. That is quite clear also when you see the spectra, right? Even if you just compare how clean and decent the spectra are in the depositional settings compared to the eroding landform positions at the different depths that I'm showing. So that was really interesting and spent a lot of time to get to this point for these papers. And then one of the other things we did was we took these soils through what we call density fractionation schemes, which allows me to distribute the carbon in soil into three pools that cycle differently. One is the free light fraction. This is the particulate residue carbon that exists in soil. It doesn't have any association with minerals. So I expect that this is the carbon that's fairly fresh and it's going to decompose quite quickly. Then the other one is the carbon that's inside the mineral aggregate spaces where the microbes, oxygen, water is having a hard time to diffuse. So it's going to be protected. It's going to persist there for a long period of time because of that physical protection. And then the third group, carbon is the majority of the carbon in these soils, on the order of about 3/4 of all the carbon in the soils, is in this pool of heavy fraction where it's actually chemically bound to the surfaces of minerals. And so you can also see here that the carbon that decomposes very, very quickly, right, is, over at depth in particular, you can see it starts out fairly similar, right, in both, but over time you could see quite quickly how it starts changing. And even if you find a free light fraction at depth, then most of it is either in the aromatic domain or alkyl domains, and not much else really makes it, which makes a lot of sense. Partly true, also a little less change, but similar to the occluded light fraction, but a much more complex picture that emerged with the heavy fraction. And this work was incredibly important and largely probably got me my faculty job and everything that you can imagine, because it's, I'm joking here, but you get the idea. It was really helpful to help us clarify, not just with numbers of this much carbon's coming in and out, but rather add the little a layer of complexity with recognizing how the chemical signatures of the organic matter is changing, how the different pools of carbon are associating with minerals and things like that, and really helped kind of clarify how geomorphology and geomorphic processes that have to do with topography of the landscape, lateral transport of topsoil, really factor in to help explain dynamics of soil organic matter, where there is really active loss of surface topsoil and associated soil organic matter from eroding positions, and then accumulation and burial and persistence of carbon in the depositional settings over a long period of time, along with those chemical signatures that we talked about. And the other piece that I wanted to quickly tell you about is fire and the kind of alterations in soil organic matter that occur because of heat and exposure to fire. Of course, what we recognize is that when biomass burns, it sometimes leaves behind char or ash, depending on the burning conditions, that is made out of condensed aromatic structures. And in our lab, we've spent a lot of time thinking about these alterations that happen to biomass and topsoil carbon in soil, in particular due to wildfires that occur in the Sierra Nevada and other places that lead to quite significant changes in soil physical and chemical properties, but also in the nature of the organic matter. And we've been really interested in using in using NMR sometimes along with FTIR to help us explain how much of the carbon remaining after wildfires is actually pyrogenic or made out of these condensed aromatic moieties. And this goes into an area that we don't necessarily have time to think about, but this is a kind of way to explain how erosion also plays really important role in determining the fate of pyrogenic carbon that remains on land after fires. Because the reality, as we've shown in a variety of different studies at this point, is that the rate at which pyrogenic carbon or fire-altered carbon decomposes is very different if it stayed up in the hill slopes where it was generated by fire versus it was actually transported by erosion downhill and deposited in riparian areas, especially in areas that are wet downhill in depositional settings. This is work that was done by Fernanda when she was a postdoc in our group some years ago to also demonstrate. So, for example, this is wood. You got wood that's going to burn in fire. And in this particular case, they were pyrolyzed under controlled conditions at 300 and 450 degrees to demonstrate basically that the more heat that biomass is exposed to, what you end up, what ends up remaining in the composition is largely aromatic groups, a little bit of others, but just largely dominated of condensed aromatic groups. And then, but the problem becomes, this is very hard to do with soil, because if you think about what's going on at 450 degrees C when biomass burns, right? When soil burns, regardless of how much carbon you started with, these are soils from the western slope of the Sierra Nevada that have a little less than 2% to all the way to 8% carbon in the topsoil. If they get exposed to heat in the lab, by the time you combust them to about 450 degrees C for about 30 centimeters, you lose almost 95% of the carbon. So now you've got an impossible situation that you can't even quantify what remains in soil. Because now we have to quantify the tiny amount of carbon left, regardless of what you started out with. But we've done our best with HF and all sorts of stuff. I'll show you. So one of the things Fernanda was doing was applying a proton NMR and adopting proton NMR to be able to be used, in this case, with water that leaches from burned soils. So if you burn soil and you leach water, because think about the fact that once a wildfire occurs, rains follow, water comes, and it still leaches through the soil. So we were trying to explain, to understand what is the nature of the carbon that is leached in dissolved form. So for this particular case, proton NMR was great. We had to do cold water extractions and then freeze dry and reconstitute with deuterium water, but then proceed with the proton NMR. And there's a lot of busy slide here, but what Fernanda was able to demonstrate quite nicely is that again, with soils that have very different carbon that I showed you again from the western slope of the Sierra Nevada, if you leach water through those soils under unburned conditions, versus if you burn the soil at 150C, 250C, and 350C before you leach them out, the differences in the nature of the extract, the nature of the dissolved organic matter that is largely pyrogenic when you reach the 350 that leaches out. And this was also just a beautiful work that allowed us to think about how NMR can actually help us expand what we could do with fire-related work. And the last piece I was going to mention was the particulate transport. So that's the dissolved transport of carbon after fires. But I already told you that erosion is really important. There's a lot of lateral transport that happens. These are pictures to help explain what I mean by that. Immediately after a fire, the landscape is full of ash and char. But you get back to the same catchment anywhere in the world, right? A few months later, or even a little over a year later, most of that char is not there, right? Because it's very easily transported light carbonaceous material. So it actually ends up decomposing somewhere else And so when you think about how fire is affecting the carbon cycle in forests, for example, it becomes really important to understand first how much of it is being converted to char, and second, how fast is it decomposing? What controls those decomposition? We worked with the National Park Service for a long time because they mapped the landscapes after fire. This was in particular after a fire in Yosemite, where they mapped the fire severity, they mapped the geomorphology of the landscape, and we located 21 plots here in areas that vary by slope as well as fire severity, because we wanted to study what makes the biggest difference for transporter char. And then we did HF demineralization plus 13C CP magic angle spinning NMR to look at the material that's transported. Now we're only focused on what gets transported out of these catchments. And what this story tells you is what gets transported in catchments that were burned at high severity but a high slope, catchments that were burned and catchments that are burned in medium severity fire but have high slope. A combination of those two variables, right? What becomes super clear is the fact that regardless of the geomorphology of the landscape, if that landscape is burned at high severity, then the material that's transported either early in the season, immediately after the fire, or at the end of the rainy season is largely made out of aromatic species. And there's not much else, basically. But in the medium severity fires, that's a different story because medium severity fires don't completely consume all the biomass, right? There's a little bit of new organic matter that starts growing. There's some remaining, and pines are known to also shed after fires. So you see a much complex picture even at the end of the growing season. This was also a result that we were able to verify using 13C and 15N stabilized stope analysis of unaltered materials that were not treated with HF. So it gives us a little bit of more confidence, if you will, of what we're saying is actually true. And the main message that we were able to get out of this, which I think was super unique, was to highlight the fact that there is this selective transport of pyrogenic, the condensed aromatics that are created by high severity fires. In particular, there's a selective transport of them post-fire. In fact, Rebecca, my former student, was able to demonstrate that 3,000 to 5,000 megagrams of pyrogenic carbon was transported from this fire in Yosemite in just the first year. And this may not mean much to you, but I'm going to tell you why this is important. Because this area, for example, part of it drains to Hetch Hetchy, the water supply system for the city of San Francisco. So if not just, fires generate a lot of erosion, so a lot of sediment makes it into these catchments after fire. Then if it's not just sediments, it's a lot of carbon that's being eroded from high severity fires, right? And specifically high amount of pyrogenic carbon, which now creates a huge water quality concern for the municipalities that have to deal with this. Because it's not just that they have to filter it. They actually, when this kind of stuff is in the water, and if they apply chlorine and other disinfection chemicals to disinfect, they create PAHs as a result, polycyclic aromatic hydrocarbons, right? That can even be carcinogenic. So it becomes a huge concern, and they want to know what kind of fires are going to lead to a lot more of this material making it into my water utility. And then I have to worry about not just the physical filtering of that water, but all sorts of other things I have to do before I apply chlorine and other chemicals. Anyway, a lot more other stuff we could talk about, but NMR is incredibly useful. It's helping us really untangle some complex questions in the soil system, allowing us to track, change that occur through natural and human caused processes is really critical for helping us refine our existing carbon and climate models. But it remains complex. It remains very, very tricky. I think a lot of methodological innovations are needed to actually get it to where I think most of us would be happy and can apply it easily. You all know these things way more than me, but some of the things that people have talked about is having to do with maybe advanced pulse sequences, dynamic nuclear polarization, improvements in signal to noise ratio, and other things that could help make NMR in heterogeneous media more adaptable, more easier to work with. And a lot of us would be super happy if we can also use 15N that like we do 13C, because nitrogen cycles, it's really interesting dynamics, but there's a lot less nitrogen in soil than carbon, and it makes it super hard. I'll stop talking here, but if anything I said, find it exciting or interesting, feel free to check out my website or the TED website, where I talk about why soil is so fascinating and amazing to anybody who would listen. Thank you all.
Thank you, Asmeret. And are there questions? Yes. Yeah.
So I just wanted to find out what kind of probe heads specifically for solid state NMR you've been utilizing for soil sciences with such matrices.
I'm going to be very coy and pass that question to our partner.
Okay. Well, yes. Well, actually, you began this work at the Pacific National Laboratory, and I was first doing work with you in 2016.
Yep.
And what we have at UC Merced is a Phoenix NMR 3.2 millimeter probe, and which we've done all the work at UC Merced with that probe.
Yeah.
Fantastic. Thank you.
Thanks.
Thank you. Really interesting stuff. So it looks like what you're mostly looking at with the carbon spectra are changes from the top and the bottom, from the aromatics to the alkyls. Have you thought about using statistical analysis to try and parse out a little bit more information from these spectra that visually is kind of tough, as you know, it's tough to figure out what's going on. We see these kinds of blobby, I'm just going to look at a fingerprint spectra in other areas of NMR. And for those, a lot of times we move towards statistics to try and pull out info.
Yeah, in the interest of time, what I did not tell you was this kind of characterization actually is a product of a molecular mixing model, a statistical tool where we plug in the different areas plus additional information on elemental concentration of carbon and nitrogen to be able to then get information on what proportion of this carbon is likely char, as opposed to just everything showing up in the aromatic region. Because when you're looking at natural organic matter, lignin is a very important component of that too. So be able to disentangle things like that, we rely on statistical tools. In fact, a couple of the speakers that are going to talk in a minute are also going to talk about the work that relies on these types of molecular mixing models that allows us to go a tiny bit step further. Yep, thank you.
Thank you. Another question? So with that, thank you, Asmeret, for a great talk.
And I will just unplug.
Okay.
Thank you.